All right, everyone, good morning and welcome to Emerald Ash Borer University. I'm Robin Osborne from Michigan State University, and along with my colleagues Anne Marie Nagel from Purdue University and Amy Stone from Ohio State University, we hope you'll find this webinar informative. Today we welcome Dan Herms, PhD, who is professor and chairperson of the Department of Entomology at The Ohio State University and does research for the university's Ohio Agricultural Research and Development Center. Dan has been involved in EAB Research since EAB was discovered in Ohio in 2002. Most people dealing with EAB know of Dan's important work in understanding and managing this pest. His research focuses on ecological interactions between insects and woody plants and their implications for pest management. His webinar today will review research that provides scientific basis for EAB management and ash conservation in urban environments. Results from multi-year insecticide trials with soil-applied, trunk-injected, and bark-applied systemic insecticides show that protection of even very large caliper ash trees is a viable option to consider as part of an integrated management program for EAB. If you have questions during the webinar, feel free to write them in the chat pod. We will be responding to questions after the webinar presentation is over to keep the flow of the presentation going. Please stay tuned until the end because we would like to get your feedback, and we will be providing a link to a survey that we'd like you to participate in. As well, for those of you needing CEUs, your survey information is necessary for us to process those CEUs. And the first 10 people to participate will receive an EAB goodie bag. This webinar is being recorded and it will be available for viewing this week at emeraldashborer.info. That recording, as well as the recordings for all the previous EAB University webinars, will be available on the emeraldashborer.info page as well. Please give us your feedback. We always want to know how we can make these things better. Thank you for attending today. And with that, I'm going to bring up Dr. Herms' presentation and we will get going. Thank you, Dan.
You're welcome. Thank you for the invitation to present, and thanks to everybody for taking time out of your morning to participate in the session today. I want to talk about some of the research that we've been doing over the last, I don't know, I guess 8 years or so to study insecticides for controlling and managing EAB in the urban forest and for conserving ash canopy as an option. And there's a few things I want to talk about this morning. I'm going to start out by addressing just a few misconceptions that are pervasive in the press about EAB control and try to dispel those. I'm going to go through the bulk of the presentation. We'll focus on results of multi-year insecticide trials that we've been conducting with various systemic products. And then I want to talk a little bit about how you might think about using insecticides as part of a integrated ash and EAB management program. I want to address an issue that seems to be coming up more frequently now, and that is EAB treatments and the implications for honeybees and honeybee health. And then hopefully this will all take about 35 to 40 minutes and I can address your questions after that. So I'll just start out with some of the key conclusions that I'm going to relay today, and then I'll kind of walk you through the data that leads us to those conclusions and kind of sum up by reviewing these. One major point that I want to make is that we found that insecticides are effective even on large trees and even under intense EAB pest pressure, the highest pest pressure. The insecticides are working quite well. We've worked with imidacloprid soil drenches, and we found that they're more effective on larger trees when they're applied at the newer higher level of rate, which also means though that you can treat less trees because of the per acre limitations on the amount of product that can be applied in a year. We found that all things being equal, that spring soil treatments with imidacloprid are more effective than treating in the fall, but fall treatments can be effective, and I'll talk about that nuance. We worked with Safari Dinotefuran and found that soil and basal bark spray treatments were providing good control even on moderately large trees, 15 to 20 inch diameter range, and that the basal bark spray, the systemic bark spray, and the soil treatments are equally effective. We found that emamectin benzoate triage provides 2 years of control even on really large trees, even at low rates. So we've been working with trees in the 40 to 50 inch dBH range. In some studies, we found up to 3 years of control on smaller trees in the 20 to 25 inch range. And in a head-to-head trial of commonly used products, we found that triage trunk injection and Zytec soil drenches were more effective than the Pointer trunk injection. So we all know what emerald ash borer is, and we all know that it's having a dramatic impact in urban areas. And as the infestations build in an area, trees can begin dying very rapidly. And indeed, people are finding, and we found, that most of the mortality occurs in just a short period of time, 3 to 4 years. It takes a while for it to build up to that level, but once it builds up to about 30% mortality in an area, then it goes to 100% mortality in just a few years. And so that's something that you need to be prepared for. You can really get caught off guard. And just talk a little bit about some of the work we've done to quantify ash mortality in southeast Michigan in forests, just to kind of hammer home that point. This shows the pattern of mortality when we started our studies in 2005. Ash mortality was at 40% in the forest, and this is mortality of trees greater than 1 inch diameter. By 2009, it was up to 99.7%. So that was 2.5 centimeters. That's 1 inch. So more than 99% of ash trees greater than 1 inch killed. And you can see that went from 40% to 100% in just a 4-year period. Similar patterns have been observed in places like Fort Wayne and cities in southeast Michigan and Toledo area. What state is that chart for? That was in southeast Michigan. That was in forests surrounding the Detroit area. We know that mortality in these forests near Detroit started in 1994 at least, and maybe even earlier, just from dendrochronology work that my group has done, and also Deb McCullough's work done. So it took a long time. Trees were dying for a long time at very low levels. And so the dotted line is kind of infers how fast it infers mortality. The solid line is what we actually documented. So it took a long time to get up to 30%, and but then it went very fast. So EAB management options, basically there aren't a lot. There's essentially, they can be crystallized down to this: either do nothing and let nature take its course, which is not an option in urban environments, because if you do nothing, that really means that you are committing yourself to removal of dead trees. Second option would be removal and replacement of living trees, preemptive removal, and tree inventories are really critical for planning preparation for identifying trees that maybe aren't in the best condition anyway and might be good targets for removing. But this can be something to consider as you think about the fact that most of the trees are going to die in a very short period of time, and if you're not prepared for that, you can be overwhelmed with the number of removals and the lack of infrastructure and budget to deal with that. Another option is sustained insecticide treatments. One of the things I'm going to talk about is the idea that maybe these treatments don't have to be continued at a high level of intensity forever. We're starting to look at now the kind of the aftermath, and I'll talk a little bit about that. And then probably what most people would consider as the most rational approach might be an integration of these. Some areas, it just makes sense to let the trees die in unmanaged areas, for example. Preemptive removal and replacement can make sense, and treatment of high-value trees can be very cost-effective and viable option. I'll just draw your attention to this document on ash conservation that is widely available on the internet, and this document talks about the idea that insecticide treatments and ash canopy conservation can be a viable option to consider as part of an integrated program. It addresses a couple misconceptions about that I'll mention in a minute about treatments and other strategies. It also points out that, given the value of trees, that studies and cost-benefit analyses are now showing that treatment can be a very cost-effective and viable approach. So two common misconceptions that we address in that document that are often used to justify preemptive removal are, one, that insecticides are not effective, and that, in fact, I just read that in the paper yesterday in an Ohio newspaper, quoting an urban forester, and that preemptive removal of healthy trees somehow slows the spread of EAB. Neither of these statements are true. We know that insecticides can be very effective if they're used properly. And preemptive removal of healthy trees, while there can be very legitimate reasons to do that in terms of an integrated strategy, does nothing to slow the spread of EAB. In fact, it can just accelerate the spread of EAB because they'll just disperse farther looking for healthy trees. But, you read these. So here, ripped from the headlines, as they say, some statements that I've pulled out of newspaper articles suggesting that insecticides are not effective. These statements are not correct. These are erroneous statements. Here's some statements suggesting that tree removal slows the spread of EAB. These statements are not correct either. And I guess the point here is if people erroneously believe these statements and are using those to justify the strategy that they're taking, then they need to understand that these statements are not correct, and that the strategy is based on misconception. So economics, cost analysis. I see Cliff Sadoff is in the audience today, and I just want to give a shout out to his work on looking at cost benefit analysis of EAB treatments, and draw attention to his EAB cost calculator, a spreadsheet model that can be used by urban foresters to analyze the cost and the benefits of different options and compare removal versus treatment and so forth. And so you can enter data from your tree inventory on number of trees, size of trees, costs of treatments, and so forth, and come up with projections about what would be the most cost-effective approach. And one of the things that these and a few other recently published studies are showing, that especially when you consider the environmental and economic benefits of mature trees in terms of stormwater remediation and other things, that it can be very cost effective to conserve the trees as opposed to removing and replacing them with small trees. So I want to talk a little bit about the work that we've done with multi-year studies of systemic insecticides, and we've worked with basically the three major approaches for applying systemic insecticides to trees, which are soil treatments. It can either be a drench or a soil injection. These are made near the base of the tree. I've shown those to be the most effective. And imidacloprid and dinotefuran are the products that are most widely used and labeled for that. Trunk injections, imidacloprid, emamectin benzoate, which is triage, and azadirachtin, which is the triazine product that we're just beginning to work with. And then the third approach is the systemic basal trunk sprays, where you apply the systemic insecticide, in this case dinotefuran, to the lower portion of the trunk, the lower 6 feet, the insecticide moves through the bark and then is distributed systemically through the trees. So questions that we've tried to address is, will they work on larger trees? A lot of the research has been done on smaller trees, and we've started to focus more attention on larger trees. Application rates, what are optimal rates? Timing, fall versus spring for the soil treatments. How long will treatments remain effective? Can you get multi-year control? And how do various products compare in their treatment? And actually went through this slide already, so I'm just going to skip over it, but I'll come back to it at the end. The first study I want to talk about is a long-term, relatively long-term study that we've been doing with the imidacloprid soil drenches. And it's a study we conducted in Toledo. We started in 2006. And the objectives here were to compare a couple different things. So here we're comparing spring versus fall, and then also a high rate versus low rate. There's two different products here that we're looking at here, Merit and Zytec, but these are both essentially equivalent formulations of imidacloprid. And so looking at spring versus fall, and then high rate versus low rate. And at the time, this 1.4 grams of active ingredient per inch was the highest labeled rate for Merit. So, since then, and based on this work, this higher rate of, double rate of 2.8 grams active ingredient is now been labeled for a number of imidacloprid products for use on trees larger than 15 inches trunk diameter. So we evaluated these treatments in a couple ways, by looking at canopy decline using this photographic scale published by Dave Smitley, and various researchers working on insecticides have agreed to use this same scale so that we can compare our results directly. And then another approach we use is to look at exit holes. And so here we prune branches from the canopy and we count exit holes. And if you prune these branches, say in April or early May before adults begin emerging, the exit holes that are present would be from last year. And then if you hold the branches for a couple months, the new exit holes would be from this year. And then you can get 2 years of efficacy data from one sampling, one pruning episode. And so these are the approaches that we've used. We treated these trees every year with imidacloprid, either in the fall or the spring, depending on the treatments, beginning in 2006. And in 2006, the trees showed no sign of canopy decline. They were healthy. In 2007, we started to see a little bit. I'll draw your attention to the white line. These are the untreated trees, and you can see that they began to decline very quickly, going from like 5% to 45 up to 85 and dying in very short order. So once they start declining, they decline rapidly. Looking at the fall treatments, you can see that those were not as effective unless they were applied at the 2x rate. At the 2x rate, they were much more effective than the 1x rate. But the 1x rates were effective when applied in the spring. And what you see is that there was some evidence of decline when trees were under peak pressure, but they still look quite good, and most homeowners would not notice 20% decline. And then subsequently, the trees have are really looking very good. And the fall trees were actually removed the following year. They declined even further, but I didn't get a chance to evaluate them. They were deemed hazard and removed by the city of Toledo. But so they've gone through 6-7 years now, and what we see is here, you can see what the study looked like when we started in the spring of 2006. No evidence of decline. They were probably low levels of infestation. By 2009, you can see the trees, that some of the trees are effectively protected and others, the treatments aren't working. And then in 2012, the trees that were not effectively protected have been removed. And so this is what the street looks like now in 2012 after six consecutive years of treatment. And so they're looking really, really good. And we think this has some implications for long-term management. What we're starting to look at now is, can we dial back the treatments? These are essentially the only living mature ash trees left in Toledo. There's some other trees that have been treated, but on city property, these are the only living trees. And so the EAB population has crashed because there's just no host material. And so the question is, do we have to treat these trees every year? Or can we do something like treat half the trees one year and the other half the trees another year? And can we dial back? Do we still need to use the 2x rate? Especially in the spring, the 1x rate has been effective. And so can we treat these trees with a less intensive regimen and a less costly regimen? One of the things that we're going to start doing this year is hang purple traps in these trees to see, if there are still EAB present. So these trees would be sinks, being the only trees. These trees would be sinks, and they would attract, the insects to trees that have been treated. And the trees can tolerate a little bit of damage. So populations are really small. Every other year may be enough. But these are the questions that we're going to start looking at now at this site. So there's a question about, are there any trees in the vicinity? There is a park in the vicinity, but as I mentioned, in southeast Michigan, where we're doing intensive studies, 99.7% of the trees 1 inch or greater are dead. And EAB is still lingering. We're monitoring individual saplings, and there are seedlings that are growing up. And so the question is, can EAB persist at really low levels? But obviously, the populations won't be the same as when there was many, many large trees in the vicinity, which there just aren't now. I don't think EAB is going to go away completely, but this is something that we'll look at. Switching gears to trunk injections, and we've been doing some studies with imidacloprid trunk injections and triage emamectin benzoate. In this study, we were interested in two questions. One, rates. And if you're familiar with the triage label, there's a wide range of rates going from low to medium high to high, very high actually. And then how long would one treatment work? And so in this study, we just treated the trees once, 2006, rather than the last study where we treated every year. Here we just treated once and then just see how long that they would work. We're working here with trees that are larger, and the last study was trees in the 15 to 20 inch range. These are trees in the 20 to 25 inch range. And what we see here is that, again, in 2006, the trees didn't show any evidence of decline. And then you can see the white line, the untreated line, how rapidly the untreated trees declined. And we injected these trees in June of 2006, just as EAB adults were ovipositing. And then we evaluated these trees in late summer, August of 2006, and subsequent years. So this is after the first year of treatment. This would be August 2006, August 2007, into the second year, and you can see no damage. In 2008, trees are still looking really good. Then in 2009, 3 years later, you're starting to see decline. And so this operates on a 1 year lag. So essentially this shows that you're getting really good control for 2 years. And then the treatments are starting to break down. And getting that really good control even at the lowest rates. And so this is, you can see it's under very high pest pressure. So under very high pest pressure with larger trees, what we're recommending now is treat every other year, and you can get dependable control. And here's data just showing from the exit hold data, confirming that the exit hold data emergence and successful larval development was also dramatically reduced on the treated trees at all rates. So here's the study site when we began in 2009. You can see the protected trees and the unprotected trees in this randomized complete block design. Now here's another study that we did not too far away. All these studies were conducted in South Toledo as pressure was building. And here we treated once in 2006. This is an independent set of trees. This is a different experiment. So we treated once in 2006, and then another set of trees we treated every other year, 2006 and 2008. And here's the untreated trees, and then you can see the pressure building over time as the untreated trees are declining. Trees that were treated in 2006, here we did get solid control for 3 years. This was at that medium rate, 0.4 I think, what was that, 0.4 milliliters. Let me back up here. So 0.4 grams. So this was what would be considered the medium to high rate on these 20 to 25 inch trees. And so here we're getting good control for 3 years, and then it's, we're starting to see some decline on average. Some trees didn't show decline, other trees showed more decline. And then here, if you treated every other year, if you treated in 2006 and 2008, you're seeing the trees are looking very healthy, even when all the trees in the city around are collapsing. So we've also worked with dinotefuran. This is in Bowling Green, Ohio, a little bit south of Toledo, where the pest pressure is not as heavy yet because it's spreading south from Detroit through Toledo down to Bowling Green. But we've compared the systemic basal trunk spray, where you spray the lower part of the trunk, penetrates through the bark and translocated systemically, versus low volume soil treatments, soil injections. So what we found here is the trees are not, the decline is not as advanced yet, about 50% decline. Based on the pattern we've seen before, I expect that to be up to about here this spring when we go back in May or June and take a look at them. But we're seeing about the same, real similar pattern to what we saw with the imidacloprid soil treatments, that the basal trunk spray and the soil injection are doing a nice job of protecting the trees. And it appears there could be a rate effect here, but it's not statistically significant. So both these rates of the basal trunk spray and the soil injection are providing equivalent control, and the trees are looking good. Now, in another study we conducted was to look at a kind of a head-to-head comparison of what are probably the most commonly used products. These are on 11-inch trees. This is the public library in Upper Arlington in Columbus, suburb of Columbus, and we compared untreated control with the pointer imidacloprid injection, with triage injection, and then the high rate of the imidacloprid soil treatment. You can see the trees are showing a little bit of canopy decline. You can see light through the canopy. If there's no decline, you can't see light penetrating the ash canopy. Here you can see light. These trees are stressed to begin with, I think, from the parking lot environment. What we found here is that, so they were all experiencing on average 20 to 25% decline to begin with. The Zytec and the triage treatment stabilized that. The pointer treatment was not effective and not statistically different from the untreated control. So that actually puts a lower level of insecticide into the tree. So here's representative trees from those two different trunk injection treatments. Now, banded ash clearwing, this is a native borer that's typically been the key borer pest of ash trees. And unlike emerald ash borer, which will colonize, infest, and kill healthy trees, the banded ash clearwing borer is a pest of stress trees, and it's a common in parking lot environments Here you can see they eject the frass. This is a good diagnostic tool. You can see this sawdust or the excrement, the frass, accumulating at the base of the tree. It's a clearwing borer, it's a caterpillar, the adult is a moth. And Lepidoptera generally, as a rule, are not very sensitive to imidacloprid. And here's a study we did looking at, we evaluate whether the trees are infested or not by how much frass is ejected from the tree. It's an index of larval activity. And what we see is that merit did not work. So the trees were heavily infested as the untreated trees. These are pyrethroid bark sprays, and this is kind of the old industry standard for controlling clearwing borers, and they were quite effective. But imidacloprid, which is generally not very effective against caterpillars, did not work against banded ash clearwing borer. And arborists who are, have been dealing with this insect before EAB are finding that they control EAB with imidacloprid, but are still having problems with banded ash clearwing borer. And so, unfortunately, it requires two separate management approaches. However, we did test triage, which it's been known to control caterpillars. It's been labeled in vegetable crops for a long time, kills caterpillars, and we've shown that it's effective against gypsy moth. We tested it against banded ash clearwing borer, and it was quite effective, just effective as the onyx, the pyrethroid spray. Onyx is bifenthrin, same active ingredient as Talstar, but formulated in such a way that it adheres to bark better. And so the emamectin benzoate, even at the low rate, was very effective on these smaller trees. You can see what size trees we're dealing with, but even at the low rate, and for 2 years, provided 2 years of control. So here is an option that will provide control of both EAB and the native banded ash clearwing borer. Now, we've been working with really large trees, and other people have too. I think Cliff Sadoff at Purdue has a study with large trees and so forth. We've been working on studies on trees ranging from 32 to 47 inch dBH, and we have 10 trees of each size, and we're treating with a low to medium rate and a medium to high rate from the triage label. And what we're finding is that it's working really well. Here's percent canopy decline. We treated in 2010 and 2012. We didn't treat in 2011, so we're basing this on the assumption of a 2-year control. We'll be evaluating this year. So these trees are looking really good, even improving a little bit, while the ash trees all around them are collapsing. Now, this is, I would consider, more of a demonstration project, because we couldn't really describe this or design it as a randomized complete block, because you just can't find these kind of ash trees growing in a row on a street like we did with some of the smaller ones. And so these are scattered over a larger area in northern Ohio, but the pest pressure is really high around them, and it's really apparent that the trees that we're treating are healthy and the trees that are not being treated aren't. So in conclusion, just to summarize what I just was telling you, that these insecticides are effective even on larger trees, 20, 25 inch trees in these various studies, and even now on these 37 to 50 inch trees. The imidacloprid soil drench was most effective on the larger trees when applied at the 2x rate, at least all things being equal. The 1x rate did not work in the fall, but the 1x rate did work in the spring. So if you treat in the fall, all things being equal, you have to use a higher rate. It seems that it's better just to store the product over winter in the bottle rather than in the soil. But if you do need to treat in the fall, you need to go with a higher rate to get the same level of control. The safari treatments are working well. The emamectin benzoate was providing 2 years of control even on very large trees, and in one study we got 3 years of control, and I think that may depend on how many untreated ash trees are around, what the pest pressure is, and so forth. And then the head-to-head comparison, triage, trunk injection, and Zytec soil drench were better on these, 10 to 15 inch trees. So a few questions. Yeah, Zytec is imidacloprid. Questions regarding red-headed ash borer, that's a cerambycid. And in Ohio, boy, the only time we see red-headed ash borer is on trees that are just really, really stressed, and trees that appear that they're going to die anyway. So we've never done any studies on controls or recommended controls. I'm sure that could be different in other regions, but that's not a situation. Dinotefuran is Safari. It's also Transect. There's a couple formulations out there. For the very large trees, how far spaced from each other are the injections placed? And so was just following the formula for the label. I kind of forget exactly what that is, but the label provides some direction for that. And regarding imidacloprid as a way to protect butterflies that use ash as a host, that's an interesting question, and I hesitate to make a recommendation for that. Some butterflies might be sensitive; they haven't been tested. On the other hand, if ash trees die, we know what that outcome is going to be. It's an interesting question. How was it determined that Safari is taken up through the bark after direct bark spray? By doing residue analyses of wood and of phloem tissue and foliage tissue, and also just through efficacy on the insects. So I just want to talk a little bit here about some management implications, and so I want to come back to this mortality curve that we documented from our work on documenting the rate of ash mortality in the forests of southeast Michigan. And as we said, the trees, once they hit like 30% mortality here, they started dying really fast. It took a long time. We know that some trees were dying as early as 1994, maybe even earlier, but that's the earliest one we found. So it took a while for it to get from here to here, but once it got to here, it went really fast. And as I said, you see the same kind of things in municipalities, which has overwhelmed foresters. And so I think there's some implications here for how insecticides can be used in managing a population of ash trees in a city. If you don't do anything, they're going to die, and people are finding that they can't cut them down and remove them as fast as they're dying, which is creating hazards. And so, this is the curve that you can expect if you do nothing. But with insecticides, you could change that curve to make it look like anything that you want it to look like. And so here are, some hypothetical examples of the infinite number of possibilities. But you can delay mortality. You can schedule the mortality out. You can delay it out here. You can see, let's see what happened to my arrow. Here we go. So you can delay out 100% mortality out to a later year, and you can delay the rate at which it gets to that point. If you want to schedule your removals that way, you can stabilize mortality, so you can cut down trees you don't want, hazard trees, trees that are under power lines, whatever, and then protect the rest, stabilizing mortality. You can prevent mortality altogether in a population. Of course, trees always die for one reason or another, so you're not going to protect trees from lightning and other things, but you can prevent EAB mortality. So, these are some things to think about, how you might incorporate insecticides into a management program for a population of trees, or if you want to protect an individual tree, that can be done quite effectively. We do get questions about environmental and non-target impacts of systemic insecticides, and so Jeff Hahn and Deb McCullough and I have addressed these in this peer-reviewed document. These are the questions that we address in here. This is easily and widely available, easy to find and widely available on the internet and at emeraldashboard.info and other sites. But if you're interested in these kind of questions, or your clients are interested in these kind of questions, then you can you can see what our thoughts are about these. I will mention the issue about bees in EAB because that comes up. One thing I want to point out is that ash is wind pollinated, and the female flowers don't produce nectar. It's not bee pollinated. It's not to say that insects won't collect pollen. The pollen's not there for very long, but we've been monitoring ash trees for about 3 years now in Ohio and Michigan, and we have never seen a honeybee foraging on an ash flower. This photograph shows, this is the bee garden just outside the honeybee lab at the Worcester campus, and these are ash trees here. And Jim, too, our apiculturist, who also has an ash tree in his yard, has never seen a bee on an ash flower. And we haven't seen here when I've been monitoring for the last 3 years. There's lots of things that are in flower at the same time ash is, crab apples, dandelions, and so forth. Here, this is at Michigan State. Here, this is our experimental ash plantation. These ash trees are flowering. There's beehives in the vicinity. And we haven't seen bees foraging here either. So there's a high diversity of bee-pollinated plants that bloom with ash. So there's a lot of things out there that bees are attracted to. This is a study showing an analysis. This is an analysis of over 114 studies conducted in Europe, not in North America, showing the most common species for which bees collect pollen. And ash is not on this list, so it's never appeared in the top 5 in any of the 114 studies that they looked at. And then this is some excerpts from a dissertation. This wasn't with ash. This was with red maple, and I guess I deleted a slide. I had a slide. Anyway, Asian longhorn beetle treatments of imidacloprid are made to red maple, and red maple is insect pollinated and bee pollinated. And this dissertation looked at imidacloprid treatments that were made for the Asian longhorn beetle program, and these were conclusions. In nectar, in red maple trees treated in New York City, no imidacloprid was found in the nectar. In pollen, there was no imidacloprid in most of the samples. In the rest of the samples, concentrations were low, 32 parts per billion for soil applications, 1.5 part per billion for trunk injections. Interesting, soil applications got a higher concentration than the trunk injection. No imidacloprid was found in nectar samples in the hive or in forager bee samples. And here's a conclusion from the dissertation that the average imidacloprid concentrations were low enough not to be of concern to honeybee health. Now, this is an insect-pollinated tree, and so I think for a wind-pollinated tree like ash, doesn't produce nectar, the risk would be quite low. Oh, here's the slide. They're just out of order. So red maple flowers, here's a honeybee foraging on red maple. They bloom earlier in the year than ash does. So with that, I just want to wrap up by thanking collaborators, urban foresters, people in the industry that have helped with these studies. Craig Shar in Toledo, Dave Beneman in Bowling Green. Without the help of city foresters letting us use their cities as laboratories, it wouldn't be possible to do these kind of studies. Also want to thank our master gardeners who are really been critically important in helping with our research and also in outreach programs. Amy Stone in Toledo, who's our EAB outreach coordinator. Without help from these kind of people, wouldn't be possible to do this. So a lot of what I talked about is in this bulletin, which is easy to find. We do need to get this updated, but essentially the conclusions in this bulletin are still very valid. the revisions would just be some incremental and relatively minor things that we've learned since we initially published that. So with that, I see that a number of questions are beginning to accumulate. So Robin, should I begin working through these questions?
Sure. I think we are starting. You've answered up to Mark Guthmiller, who wanted to know about any studies of uptake of any of these systemics on phytoplasma impacted hosts.
Right.
That's the one
So,
I have there.
Okay, so that's a good question. The phytoplasma pathogen that causes ash yellows, and we, do our studies with healthy trees. So, to the degree that a tree is stressed, you can probably assume that insecticides might be less effective, especially if the stress reduces water uptake and reduces translocation of the insecticide. Plus, the tree is just that less able to mount its, what natural defenses, and ash has some natural defense, but not very powerful defense against ash. But no, we haven't. I'm not aware of any studies specifically looking at phytoplasma impacted host plants. When injecting triage, do we have trouble with tree uptake? Sometimes we do. We don't inject very many trees in a year. I don't make my living injecting trees, so we just do a few experiments, and we can time our studies for ideal conditions, which we do. So we'll go out on a day when it's sunny and there's moisture in the ground and the tree is photosynthesizing and transpiring well, and we have not had problems with uptake. Now, under those conditions, uptake is tied to transpiration rate of the tree, and if a tree is stressed or it's dry or it's hot, uptake can be a problem. And I mentioned that banded ash clearwing study. Banded ash clearwing is a later season insect. It is active in Ohio in late July and early August, and we made those trunk injections in late July in a parking lot. And we really did have uptake, especially at the problems. At the higher rate, we couldn't even get all the product into the tree. But it turns out it worked really well at the low rate, so we didn't have to. But we waited and waited and waited under those conditions. The trees just weren't transpiring. And but with 2-year control, you don't have to time it so close. So we were making our timing just before oviposition. You could treat those trees in the spring and when they would still be protected. But yeah, I know arborists do have trouble with uptake, and this can be related to the health of the tree and to environmental conditions. Morning injections seem to help the process go faster. Yep, indeed. So trees go through diurnal patterns where they photosynthesis is much higher in the morning, and then they get mid-afternoon stress when it becomes hot. And that's a very characteristic response of trees. The state Big Tree Committee is interested in protecting 20 record size ash in the state. Do you think any of the chemical companies would be interested in donating some of their chemicals? You think any would be interested in doing some pro bono work? Boy, I can't speak for the chemical companies or the arborists, so that's something you would just have to pursue with them. Any studies on long-term effects of repeated trunk injection? So that's a good question, and was recently a study published in the journal Arboriculture and Urban Forestry by Dave Smitley and some colleagues looking at responsive ash trees to trunk injections. And what they found is that healthy trees were compartmentalizing the wounds quite well. A stress tree maybe wouldn't be as effective. As far as long-term studies, I suppose that depends on how you look at long-term. I think that study looked at over a 3 or 4 year period. I have to go back and take a look at that. And of course, with EAB, nothing is long-term because it was only discovered 10 years ago, and research has really picked up really in the last 6 years. But that's work that needs to be done for sure. course, if you don't inject them, you know what's going to happen. And if you're treating every 2 to 3 years, you know they're overlaying new wood on top of that. And so, you know, as EAB is around longer and these treatments go on longer, I'm sure we'll develop more information about that question through experience and through research. Let's see. Using arbor jet incorrectly, arborists using the arbor jet incorrectly and destroying a lot of nice ash trees by improper setting of the plugs, forcing the product into the tree. I believe they need to make a certification to use arbor jet. And so people definitely should be trained in proper use of any insecticide product. Nice blurb in The Economist about EAB in Massachusetts. Not familiar with that article. I'll take a look at that. Deb's urban slam model, are you testing it? What are my thoughts on Deb McCullough's urban slam model? Are you testing it? I am not. My understanding is she is. It looks real intriguing, what they've published, and the idea that you can protect a population of trees by treating a subset each year with a product that, like triage, that might provide 2 to 3 years of control. Looks intriguing. I'm not testing it. I think she is. Are there any studies of treatment materials getting into aquifers or well water sources? So that's a good question. We addressed that in pretty good detail in that fact sheet that I mentioned. But Rich Coles has done some work on that. If you apply this product right at the base of the tree, there's an incredible high density of roots there. Imidacloprid binds to organic matter, and then there's roots there that absorb it, that provide a powerful filter. And his studies show that it's just not going to move through that if there's even low amount of organic matter. Imidacloprid has been detected in groundwater, and that's been associated with agricultural applications on sandy soils with a high water table. And of course, on an agricultural field, you're applying it across the whole field. And in many areas, there's no roots, and if it's sandy and low organic matter, that seems to be the situation. Regarding long-term injections for most trees that you could switch off to a neonic after the population has crashed, that's a possibility, although with trees greater than 20 inches, we just don't have the data to say that, that they'll be effective. And so on, larger trees, the 25 to 50 inch trees, the only data that we have are on the triage injections. And of course, the problem is that these products are labeled according to amount of product per inch of DBH. And as the diameter of a tree increases, the biomass of the tree increases exponentially. And we saw with for example, with the 1x, 2x rate, that the 2x rate, when applied in the fall, was better than the 1x rate. So that suggests a rate effect. We know that on really big trees, even the low rate of triage is effective. And so that we see that there's an opportunity to scale up. So certainly on trees in that 20-inch range, switching to neonics can be a possibility. Thanks for the presentation. You're very welcome. It's been my pleasure. Robin points out that Deb's going to be discussing the SLAM project on April 2nd. So that that is a really interesting program. I would encourage you to tune in for Deb's project. Also watch imidacloprid around streams and waterways, negative impacts of leaf litter on aquatic invertebrates. And so the negative impacts of leaf litter on aquatic invertebrates has only been demonstrated in studies where unreasonable or unrealistically high levels of imidacloprid were applied to the tree above labeled levels. And of course, we addressed the leaf litter question as well, but certainly you want to avoid that. Many aquatic invertebrates are not sensitive, but the Forest Service is treating, they've treated several 100,000 hemlock trees in Smoky Mountain National Park, Great Smokies, for hemlock woolly adelgid. And they're finding, that the organic matter really holds holds that imidacloprid. Their risk assessment that they commissioned for those studies found that even in a worst case scenario, which would be the spill of a truck, a tank spill into a small pond, imidacloprid would have minimal impact. And so it breaks down quickly as well in water, very quickly. In the presence of sunlight, imidacloprid has a half-life of just a few hours. But we talk about that issue. Certainly it's something to be concerned about. Do you have data on concentrations of the different products and rates in the foliage? No, our data is, we've not done compare. Well, we do have some data on imidacloprid versus dinotefuran and see pretty similar patterns, although dinotefuran is taken up faster, it's more soluble. Yeah, Suffolk County, New York may have data on water table contamination of imidacloprid. I think that's Long Island, and that's where it's a situation where that's been where that has been detected in the groundwater, low water tables, sandy soils. The Johnson dissertation does address some of the water quality issues. And looks like that's the end of the questions. So if anybody has a last second question, get it in there now. Otherwise, I thank you for this. I would invite you, if you have follow-up questions, send me an email, and I'll try to respond to those as on a timely basis as I can. And thanks a lot.
Thank you, Dan. I appreciate it. And thank you, everyone, for attending. I'm going to put Dan's email in the chat pod here really quickly in case you need to send an email his way. Again, this has been great information, and I'm sure you're going to get a lot of feedback on this, Dan, and thank you very much. And I'm going to be closing the meeting here in a couple minutes, folks. So, if you have comments, just put them in the chat pod, and I'll make sure we get those to Dan. Thanks, Dan.
You're welcome. Thank all of you. Enjoy the rest of your day. And don't forget to check out Deb's program on April 2nd on SLAM.